So I can, oh, thank you, yeah. I think I have too many slides, but I think I'll ask you guys to tell me which one you're more interested. As Huang has introduced, my lab has been interested. Is it okay I just stay here?
Sure.
It's okay, yeah. Interested in using structural biology to elucidate fundamental questions in immunity. Our first love was members of the TNF receptor superfamily, and there we studied how the intracellular signaling proteins assemble into molecular complexes. And then we moved down to toll-like receptors and interleukin-1 receptors because they share a lot of similarities with TNF signaling. And really more recently, we were focusing on inflammasomes. And this will be the focus of this talk as well, but I would also like, if I have time, to show you some new data on how the transmembrane domain of TNF receptors assemble into signaling competent assemblies. So just to give you some introduction of what my lab has been doing in the past 2 decades, it is really trying to elucidate these large molecular complexes that assemble upon receptor signal transduction. And this really came out of structure studies and out of a lot of frustration to some extent, because we kept on seeing these reconstituted complexes that doesn't form dimers, trimers, or a ordered, conventional oligomeric structure. They end up showing these structures that looks like a helical assembly or amyloid assembly that we showed that we showed in TNF receptor-induced necrotic pathway, or simply cross-linked dimers and dimers of trimers that form these lattice-like structures. So really, this has followed my lab in the past at least 10 years or a bit more. And from that, in fact, we proposed that the signal transduction from these innate immune systems that we have been studying may use a somewhat different signaling mechanism. So instead of ligand binding inducing a conformational change to the receptor, followed by activation of second messengers or other signals to generate the cellular response, in the case of innate immunity, ligand binding induces a receptor clustering, which then recruit in the clustered form these adapter proteins inside the cell, as well as the signaling enzymes and their substrate, all together into a gigantic molecular assembly. So when we proposed this, the whole field was just beginning to realize that, and now this has been well accepted by the field. And we're really happy to see that. So what is the reason why these receptors need to assemble into these large molecular complexes? One way to think about this is to think about a proximity-driven kinase or caspase activation. So by bringing them together, you literally increase the local concentration of these enzymes, and then you drive the activation of these enzymes. And they also, at the same time, provide a mechanism for signal amplification through polymerization. And we and others have shown that in some cases, when we studied, these receptors become activated through a threshold response. And at the same time, maybe the threshold also helped to reduce the noise in the system. So that's basically an introduction. And you may know that this phenomenon of phase separation, liquid form of high-order assembly, that's how I see it anyway, has becoming very prevalent in cell biology in recent years. And in 2016, we tried to provide a biophysical understanding of these different kind of higher-order assemblies. In fact, we think that the liquid form and the solid form that we observe, that things that we can actually have a structure of, is really in this continuum. Between the monomer and then liquid, and then even semi-solid, hydrogel kind of structure, and then the more solid structure. So for the assembly that we study, in fact, we think we also see this transition as they assemble in cell and also in vitro. And this is certainly the case for a lot of the different systems, including RNA binding proteins that has been studied very widely. So today, I will focus on inflammasomes, which are supramolecular complexes that activate inflammatory caspases, and these are different from the apoptotic caspases that you may be more familiar with. And this include caspase 1 in human and mouse, and caspase 4 and 5 in human, and caspase 11 in mouse. So for a long time, the only known substrate for an inflammatory caspase is pro-IL-1, this guy here. An inflammasome, in fact, was identified as a entity that activates caspase 1 and then produce the cytokine. Recently, 2015, this protein, Gasdermin D, was identified independently from 3 different labs through genetic screen that this is another bona fide substrate of inflammatory caspases that had completely got missed missed by the field in so many years. It turned out this protein is extremely important for the effector function of inflammasomes. Why? This is because this protein, upon cleavage by these inflammatory caspases, this N-terminal domain forms an oligomeric pore structure on the surface of the cell. And this pore is important both for the release of the processed cytokine from the inside of the cell to the outside of the cell. It's also important for the induction of a lytic cell death. So essentially, you spill all your cellular contents to the outside of the cell. This is where I need a little bit of your input. So we have a new structure of how this NLRP3 inflammasome, which is, if you know the field, you would know that this has been the hottest target for the pharmaceutical companies. So we have a structure of this NLRP3 NACHT7 complex that is impressed. Actually, it's coming out this week in Nature. So I could talk about this, and it's a localization of the complex at the microtubule organizing center. Or I could talk about Gasdermin D and our efforts to develop Gasdermin D inhibitors. And TRPM2, our joint project on this channel that is important for inflammasome activation. In fact, it's important for NLRP3 inflammasome activation. And finally, if you're interested, I could talk about the transmembrane domain of TNF receptor superfamily. So which one should I skip? I can skip something. Yeah, it's a little bit too much. Or I can just talk fast. So as I said, inflammasomes were identified as something that can induce IL-1. Interleukin-1 is the master regulator of all the cardinal signs of inflammation, from heat, redness, swelling, pain, to fever. And one of the bona fide inflammasome disease is gout, just to put this into perspective that everyone knows about. It was initial inflammasome. The first one was identified in 2002 by biochemical purification directly from cells. And it turned out inflammasome should essentially contain a sensor protein, an adapter protein, and an effector, which is a caspase. So now many different inflammasomes have been identified, and NLRP3 is at the center of this. And this is because this particular sensor protein can be activated, I would say mostly indirectly, by a lot of stimuli. First of all, any aggregates outside the cell, uric acid crystals, amyloid fibrils, cholesterol crystals, and there's some rare disease crystals that can also activate NLRP3. And bacterial toxins, such as nitrorhizin, can activate it. Danger signals such as extracellular ATP can also activate this. And redox can activate it. So many things somehow converge to NLRP3 activation. One could think about NLRP3 as a sentinel of sensing cellular stress, if you will. And upon NLRP3 activation, it will recruit this adapter protein known as ASC. And ASC then is able to recruit procaspase 1. And these are all through domains in ASK. One is a pairing domain, and the other is a CHArT domain. I don't know if I have it here, in which they form these filamentous structures. So these are work that I summarized from a few years ago, that the single cluster that gets formed in NLRP3 signaling is shown as like a cluster of different filaments using electron microscopy. And if you further zoom in into the gigantic cluster, you can see that it's composed of filamentous structures that we can reconstitute in vitro that are shaped as a branched filament because there are two different filamentous structures in this assembly. One is the PYD assembly, and the other is the CHArT assembly. But the question, so these are all downstream to from ASK to caspase-1, but how about NLRP3 at the upstream? So we didn't know here. We didn't know about this, but this particular sensor has been pursued heavily by both academia and industry because it has been implicated in all these different diseases, and many startup companies, maybe you've heard of, has been bought for NLRP3 small molecule drugs for hundreds of millions of dollars. Although the first drug is still, I mean, the efficacy of the first drug is still a way to be seen because it's only in phase 1 clinical trial. So we started out working on this project when a protein, which is a centrosomal kinase, NACHT7, was identified as important for NLRP3 activation. So it was actually a little surprising because what would you think a centrosomal kinase is doing in the inflammasome, right? But if you look at the knockout data, it's very clear. You knock out NACHT7, you have no inflammasome activation. So we thought, let's reconstitute this in vitro. Let's see at least whether they interact or not. Indeed, they form a very nice complex between these two proteins, as you can see here. And through a lot of protein engineering, we were eventually able to image these NLRP3 NACHT7 complex and obtain a cryo-EM structure. So let me see. So this is how the structure looks like. So a lot of the inflammasomes have a domain organization as the NLRP3. They're called the NLR proteins, and they all have a ATPase domain at the N-terminal region, also known as NACHT. It's just an acronym to show this is an ATPase that can oligomerize, and followed by a leucine-rich repeat domain. So we delete the pairing domain because the pairing domain would have aggregated, so we cannot include this in our structure. So this is the NBD domain here, and then this is HD1 domain, WHD and HD2. This is the globular NASH domain. Here, not showing too well here, the movie stopped, is a bound ADP. So we can see the bound ADP in the structure. And of course, this is the leucine-rich repeat domain, and this is the NACHT7. So we could only see the C-terminal lobe, but not the N-terminal lobe. This N lobe is completely disordered in the structure. So without going through all the details, I just want to show you that we could validate all the interface between NLRP3 and NACHT7 and shown that they are important for the activation of inflammasomes on the level of caspase activation, L1 secretion, and cell death. But the question is, the structure is a monomeric structure, right? So how and why this structure explains NLRP3 inflammasome activation? So in order to do this, we thought let's look at the structure and see what we can come up with an active conformation of NLRP3. So fortunately, a few years ago, my lab and another lab from Tsinghua University solved the structure of the NLRC4 inflammasome. And this domain, this is also a so-called NLR protein. The domain organization is very similar to NLRP3. And there we know that in order to become activated, there need to be an exchange of the ADP that's bound in the inactive conformation to the ATP that's bound in the active conformation. And this conformational change is huge. You need to rotate by about 90 degrees of these two domains, NBD and HD1 domains, to generate this active structure. So we thought, is it possible that this conformational change also occurs in NLRP3? So the first thing we looked at was these genetically associated mutations in the NLRP3 protein. So in this protein, all these different, these acronyms of different diseases, NOMID being the most severe form of this periodic fever syndrome, and Markov-Wells syndrome, and the familial fever syndrome. So they all related to NLRP3 mutations, and they all map to the NACHT domain, and suggesting, and very close to the ADP binding site, suggesting that perhaps these mutations affect the conformation of the NACHT domain, and that supports a relevance of the potential conformational change in the protein upon activation. So the first thing we did was to try to make a oligomeric structure of NLRP3 in the inactive state that we observed. Then you can see we actually generated a lot of clash between the neighboring molecules. But once we opens up the structure through this putative conformational change, as we observed in the homologous inflammasome protein, now we can make a model without generating any steric clash. So this is just showing 3 subunits in the oligomeric assembly. The real thing, of course, would need to extend all the way to a full ring structure. So what was very surprising is that by looking at this, you generated another secondary interface in the molecule. So this is the important interface, right, that we already validated. But now you see another interface that may be important only for oligomerization, but now for the binding between NEK7 and NLRP3. So we thought this is great. Then we have a way to validate this model. And indeed, when we mutated this interface, both on NEK7 and on NLRP3, we see that there's no effect on the binding itself, because this is the pull down here. But this is still the binding. But the caspase activation is completely abolished. So all the mutants at this interface is shown in green here, abolished. And here on NLRP3 also abolished, validating that this could be a plausible model for the active conformation of NLRP3. So this is, here I show you a complete model of this putative 11-fold disk of the NLRP3 NEK7 complex. So the good thing is now you can explain why NEK7 is important, because it bridges neighboring NLRP3 molecules to form this inflammasome assembly.
That model accommodates the PYD domains all coming
Yeah,
together.
right, exactly. Sorry. So the PYD would have been here. Yeah, would be linked here. And that's where we imagine, that's where, which we showed in RC4, there would be a filament that coming off the inflammasome disk. So why centrosome, however, right? And is NEK7 binding sufficient for inflammasome activation? So let me yes, please.
I'm ignorant about NEK7. Is that present in all
Cells?
Cell types?
Yes. NEK7, yes, but not NLRP3.
Okay.
Right. Right. So, so there's another project that's still ongoing in the lab, which I think can fit in some of the puzzle that's still remaining from what I just talked about. So what we showed in that project is that we always observe the single puncta in inflammasome, right? why single? Why this single puncture? And it's usually localized in the perinuclear region. So what could that be? So we thought it probably is the MTOC. What else will it be, right? So then we validated this using immunofluorescence. You can see dynein, which is a pericentrosomal protein, and gamma tubulin, both supposed to localize the MTOC. And you see that they indeed co-localize with NLRP3 and ASC. We also then generated live cell probes to look at this. We have this caspase-1 molecule that's hooked up to a pair of FRET molecules, and also interleukin-1 beta also hooked up to these two fluorescent proteins. So the first thing is we can use a live cell dye. It's called CHArT. It's a taxol derivative, so it can stain tubulin, right? So again, with this, you see the puncta that gets formed, right? Completely localized, co-localized with the with where the MTOC would have been. In fact, you can almost see tiny, branches of the inflammasome puncta that also coincide with the structure of the microtubule. So really suggesting that there is a transport event that activates the inflammasome, which I will show you in a minute. So this is just a movie here. Let me see if it's running. This probably is better. So that's a single puncta that's formed in these 3 different cells. I think this one, the red is CHArT. You see the puncta form exactly there. And you can also see at the puncta, you see a decrease in the FRET, suggesting cleavage, and only at the puncta. Outside the puncta, you don't see this. So without going through all the details, I essentially want to show you that this microtubule transport through a agrosome-like mechanism is important for inflammasome activation. And this is this makes sense in light of the NLRP3 NACHT7 story, because NACHT7 always localized at the MTOC. It's a centrosomal kinase, right? So we think that this transport allows NLRP3 to meet NACHT7 at the microtubule organizing center, and that's why it gets activated there.
You said it's not the kinase
activity. It's not the kinase part. Yeah, right.
So
Yeah.
That's interesting too.
It's a scaffold, right? Because you see the kinase structure itself is bridging the subunits. Yeah. So I forgot to mention it. Yeah. The kinase activity is not required for this function of NACHT7. And the way NACHT7 works is by binding to a NACHT9 molecule. I think I don't have the slide there. And this is also where NLRP3 binds. So in fact, NLRP3 activation and mitosis are mutually exclusive for this reason. So the inflammasome cannot be activated during mitosis. It can only be active because they're competing with the same molecule. So that seems, I think, makes sense biologically. Yeah. So
any other inflammasomes require NACHT7?
No, only this one. Only this one. And this is the most. So we don't understand why these other ones. And I don't know if they have a different way to tie it with cell cycle, but this one is the NACHT7 association seems to explain at least this particular inflammasome is tied with the cell cycle, right? So, any other questions for this part? I think I'm going really fast, trying to at least. Maybe too fast, yeah.
What would happen in a cell that doesn't divide, in a postmitotic cell? Do you know? Or have you ever looked?
Does not divide. Oh, whether they can be activated. Yeah. I don't really know. Macrophage are postmitotic or not? Are they postmitotic or not? They are dividing. They can divide. They're slowly, right? Is another issue, right? Yeah. This I don't know. I don't know for sure.
Neurons.
Neurons. I don't think neurons have this, but glial cells do. Yeah. So I didn't mention, but this NLRP3 inflammasome has been well studied, in fact, in Alzheimer's disease, and maybe you know about this. Yeah, right, especially NLRP3 inflammasome. Others have shown, we can just discuss, right? Yeah, others have shown that C puncta that gets released from cells during inflammasome activation, say by glial cells in the brain, can template the filament formation of A beta. So yeah, so anti-NLRP3 drug has a potential, at least, yeah, to treat neurodegenerative diseases, and that's actually been one of the focus of the field, trying to explore that. Yeah, there's a lot of link between inflammation and all sorts of diseases. Yeah.
Is the pore forming substrate of the Cas space present ubiquitously, or in only a
it's more
subset of
ubiquitous.
cells?
It's more ubiquitous. Yeah, it's certainly in macrophages. Also, glial cells also macrophage. Epithelial cells. So all the barriers, skin, gut. So they all exist. Yeah, the pore forming. Yeah. Yeah.
Do you know which has greater affinity to bind to NACHT7, the NACHT9 or NLRP3?
Yeah, we're actually, reviewer asked us to do that experiment. My postdoc couldn't make the NACHT9 construct, so so eventually we told the reviewer we were having trouble making this construct, so we don't know how they compete exactly. But the thing is, NACHT7 is limiting in the cell, so the hypothesis would be that whichever comes first will just occupy it, and then you can't switch to it until the previous process is finished. Yeah.
Does the NACHT9?
NACHT9, yeah.
Does it bind as an independent entity, or only to NACHT7, or only if it's part of a larger complex?
No, NACHT9 binds to just NACHT7 itself. It forms this dimeric.
So is there a phenotype if you overexpress NACHT9?
Ah, good question. So we're not, yeah, no, I don't know, yeah. I only read about it. So we have not done any experiment on it, yeah. It just makes sense that there's a cell cycle dependence of inflammasome activation. Right, any other questions? Yeah, please.
Yeah, so
I don't have to talk about all of it. So by the time comes, we just finish, yeah, it's fine, yeah, please.
NLRP3 activation require ADP go to ATP.
Yeah.
So
Mm-hmm.
Is there any nucleotide activation enzyme, like we know most of the GDP.
Yeah.
Those
Oh, is there a gap?
Yeah.
It's like, or exchange factor.
Exactly. So this
Actually, that's a really, really good question. So for the NLRC4 inflammasome, the other inflammasome that we have studied, right, we actually thought that there might be something that helps it to exchange in the cell. The reason is, if we take the protein in the inactive state and purify that in vitro and add ATP or ATP analog, we couldn't activate it. So we couldn't activate it. No matter what we do, we couldn't, other people also couldn't. We actually hypothesized there might be some kind of exchange factor, but still, we didn't do any experiment to look for it. I mean, you probably had to do some unbiased screen to look for it. So we didn't do the experiment to look for it, even though we were interested in it. It's a little bit outside our comfort zone, so we didn't really do it. Yeah. For this one, we also not sure whether we can activate NLRP3 in vitro. Part of the reason is that the construct that we have has a large MBP tag at the N-terminus. So we thought there could be two reasons why when you add ATP, it doesn't form an oligomer. Maybe because the MBP is clashing, right, as you form a ring, or because you need the exchange factor. So we don't know how to do that. So we actually thought about what we can do is to take the lysate. We actually really did discuss this. We can take the lysate from a cell and just add that to the reconstitution. Meanwhile, maybe fractionate that and see which one is required. But we never did that experiment. My postdoc never did that experiment. We did Li-Man. I really talked about how, try to do that, but we never did that. Yeah.
Is there anything bound to the leucine-rich repeat in the inactive form?
Mm-hmm.
And could it be that that competes with NEC7, and that all these different kinds of ways to activate are actually removing
NEC7?
that? No.
Oh, removing that thing.
And therefore freeing its ability to bind to NEC7.
NEC7, yeah.
And that's how you could get multiple ways to.
Yeah, so we don't know that. The reason, so at least, this, all these things are out of, also outside our comfort zone, although I think it's really important to know what's bound to NLRP3 in the inactive state, active state. So all these, we need to pull it down, do mass spec and things like that. It's actually, he is interested in doing that. Long Fei, most of the people in the lab just want to have a beautiful structure. So no, I think those are actually really important questions. We have no idea.
Most people everywhere want to have a beautiful structure.
Oh, so it's hard to get people in my lab to do such a thing. Fishing expedition, right. We do know if we take the NLRP3 that we purify without the NEC7 and do cryo-EM on it, the conformation is much worse. I think it's much more flexible. The classes that I showed you looks like it has this earring-shaped thing. You don't see that in those classes. They're just blurry kind of stuff. So it's much more flexible, but whether it's bound to anything, we can't tell from those experiments. Yeah, right. Yeah. Any other questions? Yeah. So let me show you the pores, I guess. Yeah. So the pores, as I said, is important both for releasing these matured interleukin-1 or interleukin-18. So backtrack just one step. These cytokines does not have a secretion signal. So I think they're released by unconventional secretion, and that was a mystery, forever until the discovery of this protein. So now we know it's released through the pore. So how does it do that? So 2016, we collaborated with Judy Lieberman to look at the cytotoxicity of the N-terminal region. And that's when we discovered that they formed this oligomer upon activation. And when we reconstituted them in vitro, they formed these pores. So before that, nobody actually knew that's a pore. They just know it's affecting the membrane stability. So what's the requirement for forming these pores on the membrane? It has to bind to acidic lipids. So in the liposome that we reconstitute or using the lipid strip, we can show that Gasdermin D N-terminus, and only the N-terminus, not the full length and not the C-terminal tail, C-terminal part, binds to PIPs, binds to phosphatidic acid, binds to phosphatidylserine, and all these acidic lipids are confined to the inner leaflet of mammalian cell membrane. So they're not at the outer leaflet, only the inner leaflet. And that suggests the pore has to be formed from inside the cell. And once released, it doesn't harm the bystander mammalian cells, unless the cell is already undergoing apoptosis, right? You remember PS phosphatidylserine gets externalized during apoptosis. It can also bind to this lipid, cardiolipin, which is a component of bacterial and mitochondrial membrane, suggesting that it can also damage the mitochondria. Some people, these are unpublished data, but some people in fact say that perhaps the mitochondria in the pyroptotic cell is damaged even before the pore gets formed on the cell surface. So the conclusion at the time was that Gasdermin D N-terminus combine acidic lipids at the inner leaflet to form these large pores. They don't harm bystander mammalian cells, but kill bacteria. And because of this pore formation observation, the word pyroptosis that has been used for decades is now have a new meaning. So any Gasdermin-mediated lytic cell death now is defined as pyroptosis. So the Gasdermin family has these different members in human. I guess it's 6 members, A, B, C, D, E, and this member that's slightly different because it doesn't have a large enough C-terminal domain. They're known for quite some time, and they are related to these genetic diseases, but the function of these proteins were unknown really until the discovery that they can form pores on the surface. And then we and others have shown the N-terminal part of all these different Gasdermins, in fact, can insert into the membrane upon binding to acidic lipids and form pores. So that suggests that these different proteins can be activated by different kind of stimuli and induce some kind of lytic cell death. But the enzyme that does the cleavage is unknown. Only two is known. Caspase 1, the inflammatory caspases cleaves Gasdermin D, and caspase 3 and 7, which are apoptotic caspases, cleaves Gasdermin E. This is the work from Feng Xiao's group in China. So this, right here. So this, in fact, provide a crosstalk between apoptotic cell death, which are supposed to be non-inflammatory, and the inflammatory cell death mediated by Gasdermin. So we were interested in how Gasdermin can form membrane pores. Pore formation, in fact, has been a common theme of all the different kinds of cell death. In apoptosis is the BCL2 family of proteins that form pores on the surface of mitochondria. In necroptosis, this protein MLKL also supposed to damage the membrane. So membrane, it seems to be, is at the central is a central thesis for all these different forms of cell death. But all of them, the mechanism of disruption has been poorly characterized. So, student and postdoc Jianbing and Xu worked on this project, and they were able to solve this a homologue of Gasdermin D, which is Gasdermin A3 from mouse, and solving the structure using cryo-electron microscopy. So surprisingly, though, we see two rings. We were only expecting one ring. That is the cyan ring, because it has the beta region that inserts into the membrane to form a transmembrane pore. But we see two rings. And also at the time, the resolution of this thing was about 4.6 angstrom. So we couldn't really trace the entire molecule. So we thought, let's do something else. Let's treat a protein with a mercury compound to see if we can get a derivative, if you will. And this is a trick that people use for crystallography for as long as we know it. But surprisingly, once we treat the pore with a mercury compound, it disassembled the two rings. And we were able to get single rings, and these single rings went to much better resolution. So now we have a 3.8 angstrom resolution map that we were able to trace everything. So the subunit structure is shown here. It really looks like a left hand, like this. It has two beta hairpins that inserts into the membrane, and it has this palm region, and then it has a thumb, which is a alpha helix, alpha 1 helix. I will show you that this alpha 1 helix binds to the acidic lipids, and then the rest inserts into the membrane. And the entire structure has 27 such subunits. There's some stoichiometry variation. The pores that we saw can have 26, 27, or 28, but 27 is a major species. So if we compare the structure that Feng Xiao solved, this is a crystal structure in the auto-inhibited form of Gasdermin A3. So cyan is the N-terminal region, and the yellow is the C-terminal region. So the C-terminal region is binding to the N-terminal region to inhibit the function of the N-terminal region. And the helix this is the helix that later becomes the stem helix. So it's packed so that it cannot see lipids. So only upon cleavage now you can release the C-terminal region so that you can form a pore on the cell surface. So this is a morph between the inactive form and the active form. So you see one beta hairpin essentially extends to a full beta hairpin as shown here. And for the other beta hairpin, the entire structure had to be pulled into this second beta hairpin in order to form the membrane pore. On the other hand, the globular region really didn't change very much, and that includes the helix that binds to acidic lipids. So,
and do you think that happens spontaneously?
Exactly, yeah, exactly. So I think once you cleave the protein and once you see the lipids, you completely refold. It's like that region of the protein completely refold. It's a dramatic downhill energy ride. So in that sense, I think it's actually, well, I'm going to talk about our effort to look at inhibitors for Gasdermin D, but it's actually a process that's hard to inhibit for that, I think at least partially for that reason. So this is just to show you that in the cryo-EM map, we can see this little bit density that's not explained by the protein. And we can fit that fairly nicely with the head group of cardiolipine that we used in the reconstitution. So this suggests that even though, so I didn't talk about the way we made the pore. The way we made the pore is we make liposome that has these acidic lipids on it. And then we cut the protein with caspase, and then the protein will insert into the membrane. And then we solubilize this thing in detergent. Actually took JMB forever to find a good detergent that can solubilize this. He also actually almost gave up. He started using nanodisc. And during the nanodisc reconstitution, in this process, in the process of nanodisc reconstitution, cholate is the detergent that's used in that process. And then he learned that cholate actually can solubilize which is a very strong detergent. Cholate can solubilize these pores, and he ended up just using cholate. But even with the detergent, some lipids are still bound, which was very nice for us.
Those cardiolipine or the lipid stabilize the formation, or what does the lipid
I think without the acidic lipids, because which I also didn't show you, you see this alpha-1 helix is very positively charged. And this is a helix that needs some acidic group to bind to on the membrane in order to oligomerize and then insert. Right, yeah, so it's just recognition. Yeah, right. So, so biologically, there's some details that the structure helps to explain. First of all, I forgot to mention the diameter of the pore is about 180 angstrom. So L1 is pretty small, maybe about 50 angstrom in diameter. So that can go through very nicely. So that explains the pore can release L1. But how about lytic cell death? The way we define lytic cell death in our field, at least, is to measure release of lactate dehydrogenase, LDH release. And LDH is a tetramer that's pretty big, actually cannot go through the pore. So in order for LDH to get released, you need to have membrane disruption surrounding the pores. So I think the pore structure helped to desegregate live cell interleukin-1 secretion, which can happen, and that versus L1 release and everything else released through a disruption of the membrane. So, yeah.
Do you think the lipid composition might change the diameter of the pore?
So at least in vitro, we don't see a huge difference. We don't see a huge difference. So we did cardiolipine. We also used a PS, phosphatidylserine. In there, at least growth dimensions are very similar. So I wouldn't, but however, I wouldn't be surprised that there would be subtle changes. You know, maybe instead of 27 subunit, now 29 is preferred, or something like that. That I would not be too surprised.
I'm thinking if like some of the inflammatory signals may change the lipid composition.
The lipid composition,
and
yeah.
promote, like, different
Yeah.
pore size.
I think, so one thing that did came to my mind is PIPs. PIPs, you know, if it's regulated by signaling pathways, maybe it have some role in potentiating or inhibiting the pathway. But I have no evidence, I have no data on that. We do know that, you know, another, a student in Shiyu actually is also working on Gasdermin D pores. So the A3 pores are 27-fold mainly, right? The D pores have a lot more subunit, like 33 and 34. So there are differences, I think, between the different Gasdermin proteins and perhaps between different lipids. But we really need to do these things in liposomes, in nanodiscs with the lipid, with the real lipid instead of detergent. It just these pores are very large, so the traditional nanodisc does not work. You need these. You need this the belt, right, that stabilizes the nanodisc. You need what's the word? A linear, circular, linked this protein to form these larger pores. Yeah, it's a challenge, but it's on our plate to try to pursue that. So I talked about the two rings. Why do we have two rings? Remember, these proteins like to bind to acidic lipids, right? So in vitro, the liposome we make is not asymmetric. So we have acidic lipids on both leaflet of the membrane. So we think that's the reason. So it's really we think it's an artifact of in vitro reconstitution. As I show here, because now you have acidic lipids on both sides. So once you have one ring inserted into the membrane, you still have these exposed acidic lipids that can support the oligomerization and the recruitment, but it cannot insert. So by having this artifact, we were able to capture two states of the protein. One is pre-insertion state, the other is the inserted state. And we think this is a help to establish the steps in the pore formation pathway. Full-length Gasdermin first needs to be cleaved, and then once it's cleaved, in the presence of lipid, it can bind to acidic lipids and form a pre-pore. We think that pre-pore and pre-pore formation and insertion are probably concerted. They probably happen, you know, very close in time. But we can also separate these two processes by mutating residues that are important only for insertion. So anything that's in this beta tip, once we mutate that, it cannot insert but can still form this pore on the membrane. So we're also pursuing that structure as a separate entity. So we think this is essentially what happens, that once the pre-pore is formed, it can insert into the membrane to form the pore structure. So this insertion process perhaps can be inhibited, but probably also fairly difficult for traditional inhibitors.
And
Yeah.
You had mentioned that Gasdermin can be used to lyse bacteria.
Yes.
Example.
Oh,
So-
yes,
So
yeah.
then does that mean that at some point they will hop out through their own pore? Like how do they leave the cell once they're inserted into the membrane like that?
Oh, right. No, actually it's not that. So I think it's the subunits. It's the
delivered.
delivered into the periphery. Those can form new pores on the, once it's formed, you cannot reverse. You cannot, I don't think you can reverse. I cannot imagine it'll reverse, yeah. But once you have a pore, you can release more subunits, right, yeah, so that's the idea, yeah. How am I doing with time? So I think I can stop. 5:50, 5 more minutes, yeah, please.
Can the cell extrude the entire pore?
Yeah, we can just discuss. Actually, there's all these interesting stuff. Yes. So in the case of studies in perforin, perforin is this pore right, that's formed at the junction between cytotoxic T cell and the target cell. So there, after perforating, also from a pore, deliver granzyme, for example, the target cell can repair the membrane. And they repair membrane by take the entire pore out and then exclude, spit it out as an exosome. So, and this has been also shown now for Gasdermin. This is a science paper, I think, last year showed that this also happens for Gasdermin. And that's why you can have live cell secretion, because you can, how do you imagine a cell can tolerate this big pore on the surface? But you do get live cell secretion, right? So part of the rationale is that the cell can repair this so that you have a steady state of the pore. You have a steady state. You don't get too much to overwhelm itself.
And you think that's happening also in non-inflammatory, non-conventional secretion?
I think so. I definitely think so. I definitely think so. Yeah, but this is an area that has not been tapped at all. Yeah, I think so. Yeah, right. Someone should do this. Yeah, I'm sure someone is doing it. Yeah, yeah. Hey.
Yeah, so if that's the case, the secretion is one direction or both directions?
I would say both.
You can get in.
I don't see why not. Yeah, I certainly don't see why not. So one of the postdoc in the lab who has many cell biologist training, he wants to look at the secretome, what gets released from the pore. So he's gonna inhibit cell death. So he's gonna treat the cells in a condition that the cell do not die, but only secrete stuff. And then he's gonna look at what's released from the cell. Right. So we don't know. We don't know the.
But what would be like why use a pore and not like a exosome to secrete things?
Good question. I don't know. You should ask evolution. I don't know. Yeah. We actually thought that. So initially, for the light imaging study that we did, right, we saw the trafficking of NLRP3 inflammasome to the mTOC, right? So there, we also saw under electron microscopy that sometimes the inflammasome is surrounded by a double membrane that looks like an autophagosome. A double membrane looks like autophagosome. So at that time, we thought, okay, maybe interleukin-1, everything is in there, then this whole thing can just get released that way. There are still papers out there saying that, but I really don't know which way. Yeah, it just seems like this is the simplest way to do it. Yeah. Is exosome also involved? I don't know if that's a default pathway or it's an alternative pathway. Yeah, just a lot to be learned as well. Yeah.
I think about if this pore is this large, when you call something like ion imbalance.
Oh yeah. So the repair process is induced by calcium influx. So it's the calcium influx that induced the beginning of the repair process.
And how about the hypo K, the loss of K?
Also, which also,
NLRP3
yes,
activation,
it
what happens in the context?
Yeah. So if you activate Gasdermin D through a different inflammasome pathway, not NLRP3, so right, as soon as the pore is formed, you have potassium efflux, and then at that point, NLRP3 is also activated.
So
Potassium efflux also.
Can that low K be shown to activate in vitro?
Yes, yeah, in cells,
But I mean in excellular, in lysate, can you assemble the?
Oh, no. I think it's much more. I think potassium efflux is still indirect. In my opinion, it's still not the direct activator of NLRP3. It's still indirect, I think. Because long time ago, we did these try to in vitro reconstitute at least the filamentous structures, right? See if it's dependent on potassium. That was our hypothesis. So we can show in vitro it's dependent both on potassium and sodium, exactly the same way. There's no difference, at least, yeah. So I think it will still have to be indirect, yeah, right, yeah. Okay. Thank you, yeah, thank you, yeah.